Okay, hello everyone. Today I'm majorly talk about the Seurat hands-on examples for single-cell RNA-seq data analysis. Seurat is a comprehensive analysis package for single-cell RNA-seq data. So it was developed by Strategis Lab. So Seurat is good for basic data processing and presentation. So you can see it's for most of the data generated, especially the text genomics data generated as the CMG of IU School of Medicine. So the data will be always with the single-cell RNA-seq data will be always with a certain general data analysis report, which are all conducted by the Seurat 3. So however, due to Seurat 3 is optimized for the general data analysis, and also especially it has been optimized for the text chromium data analysis in the most recent version. So if you want to do more detailed analysis, if you want more detailed mechanism interpretation of your data, so definitely you may need more efforts other than the direct application of this package. Today, my purpose is to illustrate how a computational biologist may use Seurat 3 to conduct the most basic data analysis approaches to show how the data looks like and what cell clusters are inside of a single cell RNA data. So the analysis included today, including are about the data loading and quality control, data parameter checking and visualization, data normalization, data clustering and visualization, identification of cell cluster specific genes, and visualization of marker genes. So in the end, I may give some discussions of more advanced topics of the Seurat 3. So let's go directly to the hands-on part. So this is R, Windows version R. So Seurat has been deposited to CRAN. So you can directly use install.packages to install Seurat and use the library to get the Seurat code. So here is I use a const 0 as my input data, which is a general const data. So you can see that the just shows how the const data looks like. So you can see this is just the first 6 rows and five columns of the const 0 data. In total, const 0 has my data has 14 more than 14,000 of cells and more than 40,000 of genes and 1,700 cells. So each row in the data is one gene and each column is one cell. And these are the cell barcodes. It's a text genomics data. And this data is a mouse data. You can see here are the gene symbols. And we can, after we get the data, so no matter the text genomics data or SMART-SIG2 based data, if you get after you get a const matrix, we use the create Seurat object, this command to create a Seurat object. And then all the analysis will conduct to this Seurat object. So the Seurat command is more like it's written in a way in a style of the object-driven programming. So all the operations will conduct it to this this object, and the new result will be also stored in this object. So when you just tap it, it can tell you. So currently the data are with this number of features and this number of samples. So when you use this command to create it, so project name is just a name, so it doesn't matter. So and the mean cells means to include the genes that are expressed in at least 3 cells. And the main features here means the cells included when we create the data. So for all the cells in the const data, we only include the cells with at least 200 genes expressed with non-zero expression. So here you can see the dimension of the input is 14904 and by the 17772. So, but in the selected data, in the created data, we see only 40,896 features are selected, and for the cells, more around 500 cells have been removed. So due to the total number of genes, total number of expressed genes in the cells are less than 200. So here are the when we, so the even this is an object, when we click it, we cannot see anything. But when we use the names attributes, we can see what are there in the what are the features or the variables stored in this data. So here, I just want to show you that the assay and the RA in the assay part, in the assay variable, in the assay sublist of this object is actually the raw data. Here, this is the rest are majorly about the default parameters. So without specific, here we haven't done any computation to this met Seurat to our object. So most of these are empty. So the first analysis we can conduct with after we load it into a Seurat object is to compute the percentage of the gene expression from the mitochondria coded genes, coded DNA. So here is we can use percentage feature set to compute the percentage of the mitochondria genes. Due to its mouse, so all the mitochondria genes are just the mouse genes, in mouse are initialed by MT and slash, and then we can compute it as a percentage feature set, and then this may tell you for each of the cell what's the percentage of the mitochondria expression genes. So this is not limited to mitochondria, so you can, for example, you want to check all the CD markers expression, you can also use this to check the percentage of all the CD markers, CD genes. So here we majorly use it to check the percentage of mitochondrial DNA expression. So, and then we can use this violin plot to plot the three data parameters. So the first is the total number of mRNA, the number of expressed RNA observed in each cell. So we can see certain cells are with more than 7,000 of mRNA expressed detected with non-zero expression level, and due to we set a hard cutoff of 200 genes per cell, so 200 at least 200 genes expressed in the cell. So here the bottom part is 200. Here are the total counts of RNAs. So certain cells are with extremely high expression. This is highly common due to this is the text genomics data. When we construct the libraries, all the counts are put, all the rates are put together, and certain pools can be the rates from certain cells can be drawn more frequently. So this result in biased total number of counts from different cells. And here is the percentage of mitochondrial genes. Normally, we consider 10% or less than 10% as a cutoff. So due to so this has been reported before in a genome biology paper. So the cell in the single cell data analysis, if the cell are leaking, so due to the cytosol part will be leaked, and the mitochondrial part can be still there. So the percentage of mitochondria encoded DNA expression will be increased. So in these leaking cells. So normally, so we use this as a threshold to or feature to select or filter out the low quality cells. So then we can make a simple plot, two simple plots by checking the percentage of mitochondria against the total number of counts. We can see here those cells are more like high quality cells due to the total number of RNA, the total number of RNA are high, but the percentage of mitochondria are low. And on the other side, we see the total number of the cells with high percentage of mitochondrial DNA expression. The total of counts are low, thus suggesting those cells are low quality cells. And the second plot is the total number of counts against the total number of genes observed in each cell. So we see this nonlinear curve always. So, and here are the low quality cells. And based on this figure, majorly we consider the top part are the cells, are the if this is a text genomics data, we consider the top part should also be excluded due to the in the text experimental protocol. So each bit can be attached with multiple cells, so with certain probability, and that result in the total number of features or gene features observed from this bit be increased. So we consider this as the bits may attached with multiple cells that need to be removed. So in this specific data set, I use the total number of features larger than, I may select larger than 500. So based on this feature, so 500 is just around here. So, and the total number of features smaller than 6,000. So we can use 6,000 as a threshold. And percentage of mitochondria that is smaller than 10 as a criteria. So for different data set, we can set different criteria based on this how this plot looks like. So it is okay to sacrifice certain percentage of cells. So just to make sure the low quality cells are not highly contained in the analyzed data. Then we first show how the selected cells looks like. So the number of gene features are still the same, but you can see a lot of cells has been filtered out. Only 700 cells have been remained. So then we use after we select that, and we use normalization, and then we find the variable features, and we get the top 10 variable genes. Those are the top 10 variable genes name. So so the top variable genes here actually means that the genes that with the largest standardized variation that with the gene expression profile with largest standardized variation. So you can use the variable feature plot and the label points. Variable feature plot this command to plot those in this data the average expression of each gene against the standardized variance of that gene. So for these genes with high average expression but low standardized variance, so those genes are more like housekeeping genes that are with relatively stable expression through all the cells, which are less informative for the cell cluster analysis. And we consider the genes that are specifically highly variedly expressed through the cells, may more likely be the marker genes of certain cell types. So here, this data I'm using is the cells sorted with certain surface markers achieved from a tumor microenvironment. So the cells should include the T cell, majorly include T cells and the fibroblast cells. We can see here the gene expression, top gene expression, is the top varied genes are the collagen genes and also HLC, MHC class 2 genes, and also the granzymes. So those are the marker genes of the cells in that system. And then here we get kind of the method tell us so this number of genes are selected as the top varied. Top 2,000 genes are shown here. Top 2,000 varied genes are shown here. So then here we use we scale the data after we get the top varied genes, we select genes data, and then we only use the top varied features, just top 2,000 varied features to run the PCA of this normalized data. So then we print the genes associated with the top PCs, and this is I plot the top 5 genes, top 5 positively, the top 5 genes positively associated with the first PCA, and the top 5 negatively associated genes negatively associated with the top PCA. So, and also we can do it for the 10 PCs, and then this plot shows a general plot, just a general plot can be conducted by Seurat to show the top genes that are associated with each PC. And also we can use this dim plot to show the PC1 and PC2, how each data, each point is one cell. And this is just like a MDS plot or PCA plot. So, but due to there are highly varied variations or variance inside the single cell RNA-seq data. So the top two PCs may just show the difference of the genes associated with, we can see here, associated with collagen, with cell cycle. This is general T cell markers. Here, this group actually I found is the cell cycle related genes, and this is the general group of the transfer factors Zhang B and Zhang D and false. And actually those group of genes are in the downstream of HIF1 alpha, but I'm not sure. But you can see, I'm not sure if this here is indicate this is really HIF1 alpha varied expression. So, but you can see here the PC4 indicates the killer cell receptors, and also the NK nature killer NKG7 and CD8A is NK cell markers. But we only plot PC1 and PC2, we may miss this type of this variation. So then we can, after we get the PC conducted, so and this type plot, we can also plot this PC associated heat map. So here we have a parameter 500 cells used here. So basically it present this figure presents the top varied genes associated with the first PC, and the top 500 cells that with most varied expression of those genes. So you can see here are the group of, here yellow means high expression and purple means low expression. And we can see here those group maybe around less than 100 of strong fibroblast cells. Kind of very distinct fibroblast cells. And those are general T cells. You can see here are the T cell markers, but use CTLA4, maybe CD4 plus T cells, and CD8 plus T cells. And also we can plot it for multiple dimensions. So one information this type of figure can tell us is that, so for the top PCs, so for each PC, among 500 of cells, how many of the cells are with a distinct expression or non-zero expression of this marker genes top associated with each PC. So for example, in PC3, even this PC is significant, it's expressed, the variance is expressed higher than PC4, but we can see that only a small subset of cells are with high expression of this marker genes. And also it's including one mitochondrial gene, which can be caused by certain errors. And here PC4 is more general. You can see a large number of cells are with distinct expression, and this group should be NK cells marker. And for PC5, we see only a small proportion of cells are with positive expression of this side. So this is the MHC class 2 gene. So maybe indicating the antigen presenting cells. But here for the PC10, after PC10, we see a large number of purple background and very rare yellow values. So this indicates the genes, so top associated with the PC, only express in a very small proportion of the cells. So that can help you to check. This can tell us, okay, maybe PC10 to PC15 are not that distinct. So with this observation, I may prefer to select the PC1 to PC9 for the analysis. But if needed, I may also include PC12 and PC13, but exclude the PC10 and PC11. So here we only use PC1 to PC9 in the downstream analysis. So after we get, and also we can use the amble point to show the amble plot to show the loading of each piece. The variance, the proportion of the variance explained by each PC, but the problem is that, so especially for the single cell RNA-seq data with a large number of zeros in random matrix theory, there is not distribution of the of this of the variance, the proportion of variance can be explained by the by each PC. So by checking the ample point just by eyes, it may not give you a kind of very reliable cutoff. Except the ample point is very distinct. For this data, I cannot say the ample point is very distinct. So based on this analysis, this visualization, so what I may use is just use the top 9 pieces here. So then the PC use network-based class. Seurat use a network-based clustering based on the certain top PCs. So in the past they use KN, but right now they are more optimized. But I believe definitely for certain data, there are existing certain data that this method doesn't perform very well. But as a general tool, so I consider this is okay here. So then after we write for here, the cluster has been found. So we will check the names attributes of the Seurat object, and we may check it attributes and should be in the active ident with the identifier. So here are the output of the 7 classes, the cell labels, the cluster labels of each of the class, so which can be achieved by this command. So you can always go check what happens inside of the object to get the result extracted. And here are or you can use just idents to extract this. And then to visualize it, so recently Seurat recommend UMAP, and in the past it used t-SNE plot, and this is the UMAP of the results. We can see how the cluster looks like. So, and also there is we can use TC. So sorry, we should use 9. So also we can use, this is how the UMAP this is how the UMAP looks like by using 9. Just 9 pieces, top 9 pieces, and here can be outlier cells, and also we can use t-SNE plot to visualize it. So t-SNE plot may give a kind of a more distinct boundary of each cluster. So, and then Seurat offers a way to compute the cluster specific markers, because so far all the analysis we have done are all based on the unsupervised analysis. So then we want to give annotations to each of the cell cluster. So, and here is the command to find the marker genes of clusters. So this setting in the find markers, so the input is still the object, and ident1 means the first condition, and if there's no ident2 included here, it will just compare the ident1 equals to the first cluster, and then it will compare it with the cluster 1 versus all the other cluster, and um, uh, man, man percentage, man PCT actually means the minimal percentage of the marker genes expression in the first class. So it requires that the gene expression, the percentage of the genes are with non-zero expression in the first class for at least 25% of the cells are with the marker genes with non-zero expression in class 1. So with this, you can compute the marker genes of cluster 1. So we can see the top markers here, the killer markers, CD8A, porphyrin, and NKG7, and another killer marker. So this should be a NK cell, TNK cell. So due to this are all selected, the cells are all selected based on the CD3 markers and also some certain fibroblast cells there. So with certain fibroblast markers. So I consider majorly the NK cells have been excluded. So here these cells should be the TNK cells. And also there's another way to find markers. So use the addend1. The first group is addend1 is class 5 and versus a certain other group. The other group can be one or two or more. So still using this way, so you can conduct this. You get the CD7 and the CTSW, and and those genes are the marker genes of the group 5 versus the group 0 and group 3. So also you can use the find all markers command and select the only pos is true, only positive markers, and the minimize percentage of expression is 0.25, and also using a certain log fold change threshold for the to get the to get the specific to get the cell cluster specific markers. So the running of this approach is relatively slow due to it need to compute for every cluster. So if the data is very large and there are many clusters, this can be extremely slow. And also I want to mention the log fold change are not so it may not be so good for the single cell RNA-seq data due to a lot of zero expressions that are existing there. So when you compute the log fold change, so it can be biased by the zero expression. But here is how the result of the find all markers looks like. So we use the head to show it. So it should be use head, we can get it. Yes, head. So the first one is p-value, how significant it is in the first group compared to the other group, is how significant the expression of it, and the average log fold change, and percentage of expression in the first group and the percentage of expression in the rest group. This is the cluster 0 is the first group, and all the other cell cluster are the second group. And this is p-value and gene name. So also there is a way to use the find markers. So I consider the default test they use is a rock test. So you can also add certain different tests here. So this is the one concern I may have, but is so my overall point is that Seurat is very good for general data visualization and general cell cluster analysis, but you can imagine for a specific data you have. So if you are interested in specific mechanisms, for example, the cell cycle, or for example, the cell types, the identify various cell types. If you are interested in cell cycle, you should use the cell cycle genes to conduct the cluster analysis. And if you are using if you are interested in the cell types, you would like to use the known cell type markers to conduct the analysis. But for all this type of analysis, it may need certain efforts to check first the expression variation of the marker genes and also the rationality of the selected marker genes. So we can see you can implement those ideas into Seurat's approach, but the general approach of Seurat is relatively coarse. It's just for general data visualization, general data characterization. So here I may show is the differential gene expression analysis used in Seurat. Here is the differential gene expression test they are using. So what they use as did search, so Wilcoxon and Bimoda and Rock, T, Poisson, and negative binomial log, and this is the logistic regression. So all of these tests are actually wrong for the single cell RNA-seq data analysis. Math is specific for single cell analysis, and this one D-seq2 has also been tested that be wrong for the for single cell analysis, single cell data analysis. So I mean wrong is that the model is incorrect and the result can be biased. For example, of this data, because my input, the total number of cells are relatively small. So if I use must, you can see there's an error will show here. The reason is that Seurat only just direct force at first this detailed feature has been really used by the by the computer by for general analysis. Second is that Seurat directly use the default setting of mask, which is only designed for well formulated and very large data set. But for the data, especially for the cluster with relatively small number of cells in, so this may not run this run may not be, it cannot be conducted with a proper error. So, and also all the other tests are actually irrational for single cell RNA-seq data. So what we can say is that for the find markers, it can identify the top markers, but a lot of markers are biased, can be biasedly identified due to Seurat use relatively a method that are relatively easy to be conducted, but are not correct for the single cell RNA-seq data. So, but still, it's okay to show you the general how the general data, how the general characteristics of the data. But for very detailed feature extraction, especially interested in a certain data, certain gene, so definitely you may use a specific differential gene expression test to do the analysis. So here, Seurat is with a certain the visualization of gene expression in specific cell types. So this is a collagen 1A1 expression. So there are two groups of cells that are more likely because it's highly specific to fibroblast cells. So these two groups are more like fibroblast cells. And CD3E, the all expressions in CD3E. So these five groups are like the T cells, and porphyrin, so which is the cell toxicity marker, so suggests that the group cluster 1 and cluster 3 are with relatively high cell toxicity, more like CD8 plus T cell or NK cells, TNK cells. And also we can plot more. So one genes we have seen is the cell cycle markers. So we can see the group 3 is more like with high proliferation. So also we can use a slot equal to cons, and then this plot may show you the exact cons of the data. Yeah, without visualization, without normalization. So, and then here are almost all the hands-on paradigm I want to present here. So after you get the gene, the expression level of the after you get the cell cluster specific markers called, we can annotate the cell clusters, and which achieved by the cell clustering approach. Yeah, we can show here. So we can tell here is the group 6 and group 4. These are more like the fibroblast markers, and group 2 is with more cytotoxicity, and group 3 is more like with the high proliferation. So, and also we can use the markers to annotate each of the subclass. And then I may talk a little bit about the advanced topics also offered by Seurat 3. So like I mentioned, so you can see that Seurat is very good for general analysis, but in Seurat 3, they offer certain number of more advanced functions. So we can go from the weakness, weakness. So, and we see the here is the PBMC's general analysis pipeline, and it's with multiple dataset integration that transfers the label of cell types in each of the condition. But here, this analysis is with a very strong consideration, which considers that in the two systems, each of the cells have a matched one in the two in the other system. That means all the cells will be matched in these two systems. But the good thing is that, for example, if you are studying the brain tissue of human and mouse, it can help you to match the cell types in the different cell types in the human and mouse tissue. And here also they have a spatial data set analysis procedure. So text is with recently developed the spatial transforming data. So here I consider the spatial one is they need to be with kind of very detailed the data first, the input data needs to be with certain spatial information, and it can help you to identify the gene expressions that are with a specific spatial pattern. And also, this is a mouse cell atlas example, and also the multimodal analysis. Actually, multimodality is very important in single-cell RNA-seq data, especially you consider there are more different cell types. Some genes may have a middle expression in this group, but very high expression in another group. So this multimodality can tell you how the functional difference or the functional level, the quantity of the functional difference among different cell groups. And here, this type of plot called ridge plot, specifically developed by the Seurat 3, is for visualizing single genes expression in different cell groups. We can see here this group CD8, this CD8 gene is with the kind of high expression in CD8 T cells and certain other cells. But please note here, this plot, they generated an unparametric way. So for a lot of genes in the single cell RNA-seq data, so they should have a peak at 0 and a peak at with expression. So more detailed parametric tests are needed. So actually, Seurat get a very good concept in analyzing this multimodality, but the problem is that Seurat doesn't have so far. This tool, Seurat, is not encoded with a very good multimodal distribution to characterize the multimodality. So yeah, but this plot can directly tell you something, the mechanism. Yeah. And another one is this is also a case study. Yeah, and another one is for the integration of single cell attack-seq data and single cell RNA-seq data. But recently there are more mature tools to integrate this two data type. But here Seurat, the specific setting of Seurat is that it can help you to co-embedding or do the because all the analysis we have conducted for the cell clustering are called low rank representation of the data, so or also called embedding of the data, low rank embedding of the data. So it can co-embed or co-represent this data into a similar low rank space and get the cluster, the identified cell cluster be matched. So that can get you a relatively matched cell clustering inferred from the two data types. So okay, that's are all of my materials today. So here I offer in my slides you can see the detailed web pages of each topic, and also I include one YouTube another lecture that are specifically introducing the integration and the label transfer problem.